Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type your questions in for our speaker in the question and answer window on your screen. After the presentation, we will ask your questions from the question and answer window. Your questions in the question and answer window are only visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Leisha Koop from Mirus Bio.
Thanks so much, Leah, and thanks to everyone that is joining us today to talk about moving beyond PEI, improved transfection processes for AAV and LV manufacture. Before we launch into the science part, I do want to introduce Mirus to you in case you're not familiar with us as a company. One thing that I've always found really noteworthy about Mirus is our dedication to both our science and the scientists that are using our products. Mirus develops research and GMP products for gene therapy development, as well as many other cell culture applications. Our transfection reagents are novel lipid and polymer formulations that both improve transfection efficiency and reduce cytotoxicity in target cell types. And we are also committed to finding optimal formulations, optimal workflows for many different cell culture applications. Obviously, today we're going to talk about virus production, but we do have products tailored for stem cell work, CRISPR-Cas gene editing, antibody production. Obviously, large RNA transfection is very important as well. All the way down to electroporation for delivering nucleic acids with an electrical pulse rather than a chemical transfection reagent. And back to that supportive scientists bit here, our technical support team, when you do reach out to us, is composed of R&D scientists, field application scientists, and our tech team, all very well versed in our products. So we really do aim to provide that additional support that you need to make sure that when you test our products and you're using our products in your groups, you're well supported and never left on your own to try to figure things out. I'm also excited to say that Mirus is now part of Gamma Biosciences, really with the goal of enabling biopharmaceutical innovators and manufacturers to deliver complex therapies to patients more quickly and efficiently. Mirus is one of several companies in the Gamma family, including Univercells, Mirin, Astrea, Nanoprovel, and Biomagnetic Solutions. And really the goal is to offer you a workflow start to finish to touch every point in your cell and gene therapy developments, to make sure that you have the most advanced technologies to accomplish your cell and gene therapy goals. So let's talk about viral vector-mediated cell and gene therapies. Viral vectors, so very often lentivirus and adeno-associated virus, or AAV, are vehicles for direct delivery of therapeutic genes to patient cells. They are distinct, though. So gene therapy is typically the delivery of the vector itself, so very often the AAV, and this vector will encode a therapeutic transgene, so a corrected copy of an errant gene that when delivered to the patient will be able to correct those heritable diseases like spinal muscular atrophy, retinal disorders, hemophilia, and heritable Parkinson's disease. Currently, there are a few FDA-approved gene therapies on the market, including Luxturna's, or Spark Therapeutics Luxturna, and Zolgensma, which treat inherited retinal disease and spinal muscular atrophy, respectively. Cell therapy is, again, a type of therapeutic that is going to utilize a viral vector, but in this case, the viral vector is used to modify a target cell type, very often something like a T cell, and then the modified cell is re-administered to the patient. So not the virus itself, but the modified cell. And oftentimes, gene therapies are used to address cancers. Very often, they're blood cancers. And the FDA-approved therapies so far, Kymriah, Yescarta, Tecartus, Breyanzi, all treat some type of leukemia or lymphoma, but there are many, many, many different therapeutics in development to address other diseases like HIV, beta thalassemia, and solid tumors as well. And where have we come so far in this field? It's, gene therapy, cell therapy isn't novel as of the last 4 or 5 years. It's been in the making for decades. But really, the process and the support for cell and gene therapy has accelerated significantly since the introduction of therapies like Luxturna and Zolgensma. But let's look at where we started here. Often these therapeutics were developed using adherent cells, which are easy to work with. They're decent for scaling out. They are decent for developing a sufficient amount of virus to get the job done, essentially. But there are definitely some workflow issues with adherent cells and these earlier scale processes that need to be resolved as we move into the next phase of cell and gene therapy developments, where the number of diseases being looked at is significantly higher. The average dose per patient also is higher. And of course, we're now looking at addressing diseases with significantly larger patient populations. So no longer just orphan diseases in small populations. We're hoping to be able to accomplish much larger diseases. And so for this, we need to move beyond what has been historically okay for viral vector production. We see a huge shift towards suspension culture because it's much easier to scale up. We need more. We need higher process yields. We need more functional viral particles per mil of culture being run. And of course, this all has to drive cost down so that we can deliver more therapeutics to more patients and not have it be crippling financially. And this is, of course, where we stepped in to try to address these types of problems. So Mirus very deliberately developed the transit virus gen transfection reagents, not only for the support, but also with quality in mind. And of course, that support component, again, for ensuring you have access to scientists that can lead you through viral vector process development so that you can attain those lower cost goals, higher production. And so this is transit virus gen. It is a very unique non-liposomal solution composed of cationic lipids and polymers that we designed very specifically for enhanced delivery of multiple different plasmids to HEC293 cells for LV and AAV production. So what we want with this reagent is that superior performance, so better delivery, better expression of functional viral particles. VirusGen is also quite flexible. So there are probably thousands of different HEC293 cell types in the world, and they certainly aren't all the same. But what we have found with VirusGen is that it works very well in a broad range of HEC cell types, whether they're adherent or suspension. Of course, you can expect that batch-to-batch reliability, and it's also a worry-free reagent for you. And what we mean by that is there's no commercial license required for R&D or manufacturing use, so no reach-through licensing at all. It's chemically defined and AO-free, and it also is available in GMP formats. We also offer the VirusGen AAV and LV kits. So these kits contain the transfection reagent I just talked about, as well as enhancer components, which will often increase titers 2 to 10-fold over using transit VirusGen transfection reagents alone. And the kits are also available in RUO Select, which I'll explain in a bit, and GMP configurations. So of course, your likely first question is, how does VirusGen perform against the PEI-based reagents that have been historically used in cell and gene therapy for the last decade? Is it worth testing? Because we know these reagents, this is new, how are we doing in terms of performance? And so we'll walk through that before we walk into the optimization part of this, just so you understand what you can expect with the VirusGen suite of products here. So in this experiment, we were generating AAV2 in XB293 cells grown in XB media. So another thing you'll notice in cell and gene therapy is there are lots of different cells and medias and different combinations that you can use. And so we'll try to address a few just so you can see what has been tested and perhaps options for you to consider as you're looking at your own workflow. But what we see here is Transit VirusGen. The bar is the black bar. VirusGen AAV system is the red bar. And we're comparing to two different PEI reagents that are offered in GMP grade. So what we see with VirusGen alone for AAV production here is about a 4 to 12-fold improvements over the PEI-based reagents. And then when we look at the full VirusGen AAV system, we can attain 8 to 24-fold over the competing PEI reagents. So definitely an advantage to test both the reagent and the enhancer. And here we're looking at lentivirus production using the VP293 cells and LV max growth media. Cells were transfected at 4 million cells per mil using 1 microgram of DNA per mil of culture. And again, we're seeing a nice improvement over the PEI-based reagents with just the Transit VirusGen reagents. But when we incorporate the LV enhancer along with transfecting with VirusGen, we get a 9-fold improvement in functional LV titers over PEI. So now let's talk about optimizing your specific LV and AAV workflows. So there's probably a lot of teams here that have already established workflows. They're comfortable with their cells, their media, their DNA, but there are also a lot of teams that are just starting out, and it's a bit overwhelming to try to decide which cells you want to use and where you even start to build your workflow. So we'll walk through some of those key parameters here. One thing that we will always look at when we're tinkering with a different HAC293 cell type is the cell density at transfection. Some cells are definitely happier at a thicker density. Some don't so much prefer that. And so it's something that I would highly encourage anyone to test when they are looking at optimization. Here we are again using XP293 cells and XP media for AAV production. The gray bar is VirusGen alone, red bar is VirusGen plus the enhancer. And if we look at just the gray bars, we see that the max titer is achieved using a cell density at 2 million cells per mil. But when we incorporate the enhancer component, we definitely see a nice uptick in titer when we shift our cell density to a little bit higher, to up to 3 million to 4 million cells per mil. So our protocol with the enhancer will typically center around about 3 million cells per mil. Fairly similar story for lentivirus production. Here we see, again, max performance at 2E6 cells per mil when just using the reagents. But when we're using the LV enhancer component, having more cells is definitely beneficial, almost in a linear fashion, where we see significantly higher titers at 4 million cells per mil. And in this case, we transfected freestyle 293F cells grown in freestyle media. One other thing to note is we used a 3-to-1 ratio of reagent to DNA and 1 microgram of total DNA per mil of culture, regardless of the cell density. So we didn't scale DNA per million cells. We maintained the same DNA amounts even when moving it to 4 million cells per mil, because there's always an excess of transfection complex particles. Often there is not a need to scale DNA linearly with cell density. So you can typically increase that and not so much have to worry about increasing your total DNA. Another very important thing to look at when optimizing transfections, especially with reagents that aren't PEI, is of course, the cell density, but also the reagents to DNA ratio, which means the amount of transfection reagents in microliters to the amount of DNA in micrograms. And in this study, we looked at AAV2 production in viral production 2.0 cells grown in AAV max media. And we also looked at 3 to 1.5 and 3 to 2 ratio of reagent to DNA. So if we focus just on the left-hand side, we're looking at VP 2.0 cells transfected at 2 million cells per mil. And what we see again is that really nice boost when adding the enhancer component, so 2 to 3-fold over VirusGen alone. But one other thing that we see is that we can actually decrease the DNA amount a little bit. So 1.5 micrograms per mil of culture seems to be perfectly sufficient. There's really no advantage to increasing DNA here. So when building that overall workflow, that's definitely advantageous and a cost savings component when thinking about the cost of GMP grade DNA. On the right, is basically the same experiment, only the cells were transfected at 3 million cells per mil. So again, we see that nice increase in functional virus titer when we do increase the cell density, and the conditions repeated as well, where the 3 to 1.5 was just as good, if not a little bit better than 3 to 2. So definitely worth doing that titration of reagent to DNA to make sure that you've got what's right for your cells, media, and vectors. And just to kind of hammer home that ratio and total DNA point, in this experiment, we transfected viral production cells, the VP 1.0 cells grown in XB media, again for AAV production. And here we're looking at even more ratios of reagent to DNA. So on the extremes, we're looking at a 2-to-1 ratio using either 1.5 or 2 micrograms of total DNA. In this case, we didn't use the enhancer at all, so the complexes were just formed in PBS, 2 million cells per mil for transfection. And if we just looked at these data points, we would say, well, they're pretty equivalent. There's not a huge advantage to shifting to 2 micrograms per mil. Really no need to do additional optimization. However, if we do look at additional ratios, so look at 1-to-1, 1.5 to 1, 1.75 to 1, we see that we can almost double our titers if we do that titration and get a better understanding of what ratio is optimal for our specific system. So here we would determine that 2 micrograms total DNA, 1-to-1 ratio is really a perfect fit for this specific expression system. And the last parameter that I want to highlight here, and of course there are many, many more we could discuss, so of course we're open to having those questions at the end, is cell culture media. There are a lot of them on the market, and we've tested quite a few at Mirus. Here we're looking at a comparison of XB cells and XB media versus viral production cells grown in LVMAX, Bricel F17, XB293, and the balanced CD Hec293 media from Irvine. The previous ones were all from Thermo. And what we see is really all of these medias are quite good. They're quite supportive of cells. We see good expression. We do tend to see higher performance pretty consistently with the balanced CD media from Irvine, so definitely one to check out. And though we're not going to show the data, we've done basically this exact experiment for LV production in the same cells, and we see very similar trends. So all of these medias are really quite good. Some do tend to be a bit better than others, but again, test them with your cells and make sure that this aligns. So until this point, the data that I've shown you was all infectious viral particles, so transducing units per mil of culture, where we determined the titer by essentially making virus, harvesting the virus, and then transducing target cells to look at expression of our transgene. So that's definitely a very solid way to titer your AAV and LV stocks, but it is a long, fairly laborious method of determining titer. So when we're talking to customers, we find that many will look at qPCR and ddPCR to determine the genome copies per mil of culture rather than doing the infectious titer, or they'll use an ELISA to look at viral proteins, so capsids per ml. And all of these methods are definitely good and an excellent way to determine a part of the viral system. But qPCR and ELISAs aren't going to tell you the full story, which is why people tend to look at both of them and then look at a ratio of the two to determine the percent full. And so we'll walk through a little bit of that in these next few slides. Here we're looking at full and empty AAV particles, again, AAV2 here. And what we observe when we're looking at titers using ddPCR versus ELISAs is really a very different trend. So we're comparing VirusGen, VirusGen Plus Enhancer using two different vector sets, so vendor A, vendor B. And what we would, if we only looked at capsid quantitation, we would assume that results with vector set A were better, were superior to vector set B. If we only looked at ddPCR, we would have the exact opposite take. So different trends are observed for physical and capsid titers. However, when we look at functional titers, and percent full capsids, and even the genome copies, we tend to see a pretty similar trend, which is nice. So this confirms that functional titering is certainly a good way to determine, both your the amount of virus that you have in the prep, as well as how effective it's going to be when transducing target cells. And we also did, again, that ratio of genomes to capsids to determine the percent full. And so when we look at all of these titering methods, the take home from both the functional titering and the quantitation of intact viral particles is that vector set B definitely tends to give us better results. And we can definitely trust our functional titering results to give us a good picture of what's going on with the virus prep. Unfortunately, not every type of virus is great for functional titering. So AAV2 transduces cells in vitro really well. So you can definitely functionally titer that. But here we're looking at a physical, so genome copies per ml on the right here, and those would be the dots, versus the functional titer, which would be the bars. And if we look at just AAV5, we see that we have a really strong physical titer. In fact, it's higher than all of the others, so AAV2, 5, and 6 here. But when trying to transduce cells in vitro, the result is really quite small. And this is very well known with different AAV serotypes. AAV5, 6, 8, 9, none of them are really great for transducing cells in vitro. And so functional titering may not be the most effective way of measuring your results. So let's kind of zero in on AAV6 here, where we're looking at kind of a blown up version of the titers. So we're also looking at capsid titer here, which is the new graph that showed, and we can also see the physical titer as well. In this case, the ddPCR results and the capsid result really aligns nicely. And then when we look at the ratio of genomes to capsids, we see that with VirusGen, we get about 50% full viral particles. And with the full AAV kit, we see about 59%. And it's cell culture, so this is probably really within error. But we see a very nice percent full using VirusGen in this expression situation. So the last few slides, I want to focus on questions we often get for teams that are scaling up for GMP or large-scale manufacture. One of the first questions that we'll always get is, how long is your complex formation window? Do we have to have everything, all of the complexes added to cultures within 30 minutes, within 15 minutes? Do we get an hour or two? Really, what amount of time do we have to work with when we're establishing our large-scale workflow? And in this experiment, we looked at AAV2 production in XB cells and XB media. And what we find is that with and without the enhancer, we really have a pretty robust complex formation window. So I should note that this isn't a log scale. So, small changes will be noticed here. But what we would kind of conclude is that anywhere from 30 to 60 minutes is a really good window for virus production. If you need to decrease that, that's fine. If you need to go a little bit longer, that also tends to be fine. So it's a good, robust window for getting complexes made, incubated, and added to cells. Another question that we get is, how do you form complexes when you need to do a 10 or a 20 or a 50 liter transfection complex? And what we tend to recommend is first combining your plasmid DNA in with a complex formation solution, and then doing just a slight dilution of the Transit VirusGen reagent, really to just make sure you're not losing reagent sticking to plastics, and then combining that with the DNA, mixing it really well. If you're doing this in a wave bag, you can just increase the angle rocket for about 5 seconds. That really is sufficient for getting complexes mixed together well, and then incubate stationary for 15 to 20 minutes, and then either pump or drain complexes into the bioreactor. We've got teams that have run this exact process well over a dozen times, and it has not failed yet. So it's definitely a very robust process for large-scale transfection. So to really kind of summarize why that's important, why is it important to have higher titers, a better process? Really what it boils down to is higher titers equal lower costs of goods for that whole transfection run, which enables you to develop more patient doses in a much more effective, cost-effective process. And we'll wrap things up with VirusGen here. I noted before that it is available in research grade, select grade, and then GMP. And what that really means, so it's exact same formulation across the board, same performance, but just different quality release testing and GMP. Everything that touches our GMP reagent from the manufacturers, final fill and finish testing is all GMP certified. So definitely compatible with those large scale workflows. And then last slide from me, which is just a summary of VirusGen. So what we wanted was a transfection reagent and systems for significant process improvements for manufacture of LV and AAV. And that is exactly what we were able to obtain with this product. So efficient across HEC293 cell types, whether they're adherent or suspension, very high performance, and excellent support from our scientists to make sure that when you test, things go smoothly. And if you are interested, we definitely want you to talk to us so that we can set you up with a sample for evaluation. And so now I'm going to hand things back to Leah for a couple polling questions before we do Q&A.
Okay, great. Thanks, Leisha. So there are a couple of questions on your screen. If you can go ahead and use your mouse to select the answer that best applies to you. The first question is, what are your biggest hurdles when developing a large-scale viral vector production platform? Please select all that apply. Scalability and expression variability, inadequate viral titers, cost of materials and labor, meeting accelerated timelines, and regulatory requirements. And the second question is, there are many HEK cells in the world. Why did you choose your specific HEK 293 cell type for LV and or AAV generation? Please select one. They generate the highest titers, regulatory support and GMP compliance, or GMP availability, licensing and or cost considerations. They're what we have in the cryo tank, and I don't use HEK 293 cells. So you can go ahead and select the answers that best apply with your mouse. We'll get started with the Q&A, and you can go ahead and enter your questions at any time for the Q&A as well. So we do have our first Q&A question, Leisha, so we'll just go ahead and get started. What is the largest scale transfection that you performed with VirusGen, and how do you recommend mixing complexes?
Good question. So at Mirus, the largest scale that we have run is in bioreactors, just 2 liters, and we tend to mix in bottles. So really simple, but we have customers that are doing transfections at 200 liters, and we worked very closely with each of these teams. Some in the CDMO space, some in medical research, some in pharma. People develop their own complex mixing process based on, some of the requirements of their system. Some absolutely everything that goes into the complex for a 200-liter reactor has to be filtered. And so we work with them on that, where reagents can be filtered before it's diluted. So you work on that process. Obviously, dilute DNA and complex formation solution first, filter that, pump the reagent in via filter, wash the filter, mix that way. It could be, if it's small scale, mixed by inversion. If it's larger scale, again, wave bags work really well, or even much larger bottles or stir tank reactors. As long as you mix thoroughly, which doesn't take that much, and then let them incubate static. So don't mix during the complex incubation period. All should be fine.
Okay, great. And what is the largest scale you have performed transfection at, and have you analyzed full to empty ratio with the AUC?
Great question. So we have not analyzed full to empty with AUC, and I definitely recognize that would be an excellent thing to do, and we are working on getting that done. But at this point, we're kind of reliant upon the same things that a lot of our customers are, which is the ddPCR and ELISAs to determine GC per ml titers, capsids, and then doing the ratio. So again, largest transfection we've done at Mirus is 2 liters in a Sartorius bioreactor. Largest customers have done, we've quite a few at 50 liters and quite a few at 200 liters as well.
And have you looked at the transfection efficiency using the system?
Yes, and so transfection efficiency is an interesting topic because high transfection efficiency doesn't necessarily mean good virus production, but it is required for good virus production. And so what we expect to see with VirusGen is at least 80% transfection efficiency. So that would be, say you're transfecting in just a single plasmid with GFP, or GFP happens to be a component of your plasmid matrix. We would expect to see at least 80% of those cells be transfection positive or expressing GFP. If the transfection efficiency is low, say it is 30%, there's definitely something going on with the culture system that we should talk about. It could be a component of the media. It could be that your cells are not in the right growth phase. So we expect high transfection efficiency, and if that's not the case, we need to talk about it.
Okay, so it looks like we have time for just one more question. If we didn't get to your question, we will pass those on to Leisha, and she can follow up with you directly. So please feel free to continue to type in your questions. We will pass those along. But the last question for the webcast today is, have you developed an assay to quantify the amount of VirusGen and AAV preps following purification?
Yes, that I would say is probably the number 2 or number 3 question that we get from pharma and CDMO teams is, do we have a residual reagent assay to make sure that there is no ancillary material in the final drug product? And we do have that assay developed, and we can definitely discuss that more at length, between customers and Mirus, to make sure that you have access to that assay.
Okay, great. Thanks, Leisha.
All right, thanks everyone for joining.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.